Hello and welcome back to HSC Sylvie. My name is Minji Kim and today I'll be covering cell membranes. Before we go into the cell membranes a little more, let's do a little brief review of cells and what we know about them. Cytology is the study of cells, and cell theory has three parts. Number one, all organisms are made of one or more cells. Number two, the cell is the most basic unit of structure and function of all organisms. It's like the atom of the elements, if you have taken chemistry or know a little bit about the periodic table, and chemistry in general too. Alright, and number three, all cells come from other cells. Those are the three parts of cell theory. Another thing to note, all cells, both prokaryote and eukaryote, have cell membranes, cytoplasm, genetic material, and ribosomes. So this is all cells. Alright, and then let's do a quick review of what a prokaryote is. A prokaryote has no internal compartments. Its only organelle is the ribosome, and it has no true nucleus, which means the genetic material, which can come in the form of DNA or RNA, is just kind of floating around in the cytoplasm within the cell. Most prokaryotes are simple unicellular organisms, and they also reproduce through mitosis. An example of a prokaryote would be bacteria. Eukaryotes. Alright, so eukaryotes have several internal structures and organelles, including but not limited to a nucleus, ribosomes, Golgi apparatus, etc. They contain actual true nucleus and they have chromosomes. They reproduce either sexually or asexually, so meiosis or mitosis. And they are either unicellular or multicellular. So a unicellular example of a eukaryote would be yeast, and a multicellular example of a eukaryote would be plants, fungus, us, animals. Alright, and that is the basic overview of cells. Let's dive right into cell membranes now. So, all cells have plasma membranes. Plasma membranes is just another way of saying cell membrane. Okay, in eukaryotes, the cell membrane is found around the cell, nucleus, vacuoles, mitochondria, and chloroplasts, and they make up the structure of the endoplasmic reticulum and the Golgi apparatus. The cell membrane functions in cells to control what enters and exits the cells for homeostasis, or internal balance and stability. It also protects these cells and also supports these cells. So cell membranes are very, very important to the continued existence of life in all organisms. Let's look a little more into the structure of the cell membranes now. So the cell membrane is made up of a phospholipid bilayer. And if we think about phospholipids, we have to think about, this silly little thing with a ball on the top, which is the phosphate head, and the two tails. And here we see, here we go, hydrophilic head group and hydrophobic tail. So the head, or like the little ball here, that's going to be a phosphate head, and it is hydrophilic, so it's attracted to water. And then we have the fatty acid tails of a phospholipid, and they are hydrophobic, which means they dislike water and they want to get away from it as fastly as possible. And it's very interesting because bi means two. So essentially how the cell membrane works is that there is a phospholipid bilayer, so two layers of phospholipids as shown here, with the fatty acid tails on the inside and the water-loving heads on the outside, because there's like water surrounding both sides, and the hydrophilic heads want to be with the water, and the hydrophobic tails do not. Hence, it makes a phospholipid bilayer. And then within this phospholipid bilayer are embedded proteins and strengthened with cholesterol molecules. So here we can see some proteins, we can see cholesterol, and we can see carbohydrates. So the membrane exists in the fluid mosaic model. It's called the fluid mosaic model, fluid, because the proteins are constantly moving or flowing within the lipid bilayer, so they're not like set into a specific place. They can move around. It's called a mosaic because if you've ever seen a mosaic, it's made of a bunch of different tiny parts. And it's kind of like that here with the proteins and carbohydrates and the lipid bilayer. And the cell membranes have pores, and these are selectively permeable, and selectively permeable means that it allows some molecules in and keeps other molecules out. Alright, proteins and carbohydrates. Here we can see like a little bigger that we have a phosphobilipid. Again, we have proteins embedded in here, and this is a channel protein. And we have little ID tags that identify, oh, this is this, and these carbohydrates essentially help distinguish the cell and say, oh, it's naturally supposed to be here, or it's not supposed to be here, and also just essentially acts as an ID tag. Alright, let's talk cell transport. Cell transport is defined as the movement of substances across the cell membrane. And there are two types of cell transport, passive and transport. Passive transport and active transport. Oh, and look at that. I spelled passive wrong. It's okay. Please ignore that. It's passive, as shown in this diagram. Alright. Within passive transport, there are two types. There is the simple diffusion and facilitated diffusion. Passive transport in general requires no energy, so you will not be seeing any energy or ATP being used in passive transport. In passive transport, the molecules kind of move randomly, but they do move with the concentration gradient. So it means that the molecules will always go from a high substance concentration to a low substance concentration. If there is an equilibrium between the both sides with the substance concentration, the molecules will not move. So in simple diffusion, we have smaller molecules and substances going directly through the phosphobilipid layer, as you see here. And it's generally for gas or liquids, and they require, again, a concentration gradient. They have to be neutral, so they cannot be ions. They cannot have a positive or negative charge. It simply will not work. And then there's facilitated diffusion, which is, again, a type of passive transport, so it requires no energy. However, in this case, we do need those integral proteins, the embedded proteins. And so we see a carrier protein and a channel protein. So the protein size and shape can determine what exactly it's letting through. So essentially, they're kind of like guard dogs that allow some things through, but not others. Again, it needs a concentration gradient, or else there simply will not be movement. And they transport bigger substances and ions, so ones with charge. Okay, and then let's go on to active transport. Active transport, you don't necessarily need a concentration gradient. You don't need it. However, in active transport, we do use energy. So there does need to be the use of ATP. Again, reminding you that ATP is essentially the currency in energy for cells. So we see that active transport is needed when we want to transport things against the concentration gradient. So with the concentration gradient would be high concentration to low concentration. Against the concentration is going to be low concentration to high concentration. So the proteins are activated by ATP, and it causes the protein to change the shape and essentially kind of scooch the substances through and against the concentration gradient. Here we see diffusion in a little more detail. So here's the lipid bilayer or the cell membrane, and then this is like extracellular means like outside, intracellular means inside. So we see that there's a concentration gradient. This side has many more of these molecules on the outside than there is on the inside. So these molecules, being small and able to fit through the lipid bilayer because they are neutral and tiny, they can go from high concentration gradient to low concentration gradient. So it's going this way. And then it kind of evens out, and when it evens out, it's going to there's no longer going to be any more movement, because why would you move? There's no need to move. Both sides are balanced. Okay, and then here's facilitated diffusion for larger molecules, and it uses proteins to enter and exit. Facilitated diffusion, so again, passive. So no use of ATP, which is, again, currency. ATP is amazing, and you should probably go research it a little more, and it's very interesting. Okay, and then there's endocytosis and exocytosis. These are examples of active transport, and here it's being used to bring in molecules in bulk and also expelling waste. So this would be for things like hormones. It's when you can see that it actually makes a little vesicle where it literally just kind of makes the little pocket of whatever it wants to bring in or like scooch out, and it kind of conforms around it, and then it goes through the membrane. So we have endocytosis, which means it's bringing stuff into the cell. Right. Then there's exocytosis, which would be the opposite, where you bring out stuff from the inside to the outside. So that was a really brief review on endocytosis, exocytosis, diffusion, and facilitated diffusion with both active transport and passive transport. And I hope that you guys learned something, and I hope you also go out and research this a little more, because it is very interesting, and I believe that you should pursue your passions. Alright, have a wonderful day. Thank you.